Welcome to this next video in which we are discussing the visual system. Okay, so in this next video what we're going to discuss is the structure of the retina. We're going to have a look at the cells that make up the retina. Now we're not going to have a look at absolutely every single cell that you can find within the retina. We're going to look at the basics. We're going to look at the most important cells. So we're going to study the photoreceptors, the rod and cone cells. We'll also have a look at the retinal pigment epithelium, and then the bipolar cells and the ganglion cells. Okay, so the way I'm going to do this is I'm going to draw out a great big picture of the retina, and we will start with a portion of non-foveal retina. So one of the big things that I want to stress to you is the difference between foveal retina and non-foveal retina. But we'll start with a picture of non-foveal retina, retina that isn't within that fovea pit, where we know that the retina is going to be much less thin. Okay, and we'll start with the outer portion, the bottom of the retina, the portion that's in contact with the choroid underneath. So I'll start this picture here. So we're going to start then with the retinal pigment epithelium. So the bottommost portion of the retina is the retinal pigment epithelium, and below the retinal pigment epithelium you'll have the choroid. So I'll label this on here. So I'll colour the choroid in pink here. So this would be where the choroid is underneath the retina. And now we're going one layer up from that, so we're now going up to where the retinal pigment epithelium is. So we'll have a basement membrane here, which I'll colour in green, and sat on that basement membrane we're going to have the retinal pigment epithelial cells. So I'll draw these on here. And we'll see the function of these cells in much more detail later on. Their function is in regenerating the photopigment, regenerating what we will see later is called 11-cis retinal. And another function of the retinal pigment epithelium is in absorbing any light that comes through the rest of the retina to this bottommost portion of the retina and stopping it from reflecting back up, because it would be a disaster if light came through, wasn't absorbed by the photoreceptors and then ended up being reflected back, because then it could be absorbed by the photoreceptors again, and that would mess up our view of the world, because the photoreceptor wouldn't be able to tell the difference between light that's coming in and hitting it straight away and light that's being reflected back. So we don't want light to be reflected back because it would mess up the image. There's also the problem that if light is going to be reflected off, then it could go off in a completely different angle and it could activate photoreceptors at inappropriate places on the retina, and that could mess up the image as well. So overall we do not want light reflecting off the retinal pigment epithelium and going back up and activating photoreceptors in the layers of the retina above the retinal pigment epithelium. And that's one of the functions of the retinal pigment epithelium, to absorb light and prevent it from reflecting back up. So here we go, this is the retinal pigment epithelium then, often abbreviated down to the RPE, but I'll write its name out in full just once. So retinal, meaning pertaining to the retina, pigment epithelium. And it appears very dark, the retinal pigment epithelium, because of the fact that it absorbs light. So remember, if something appears black, it means that it's absorbing a lot of light. It's not going to reflect the light off, otherwise it would appear coloured. Okay, right, so the retinal pigment epithelium appears very dark. Whoops. So I'll underline it here in the darkest colour that I have, which is this blue-green colour. Right, so now let's go up to the layer on top of the retinal pigment epithelium, and here is a curiosity that you will have to get used to if you haven't seen this before. So you may well have heard about this before. If you haven't, this might come as a bit of a shock initially. The portion of the retina on top is kind of upside down from what you would expect. You would expect right at the top of the retina, i.e. the portion of the retina that's actually going to be hit by the light first, you would expect that to be the place where the photoreceptors are going to be, the cells that are going to respond to light. And in fact, I'll just write that keyword down. So this word photoreceptor, this means one of the special cell types that is capable of responding to light. So this means a rod cell or a cone cell. Okay, you would expect, if you were going to design the retina, you would have expected the photoreceptors to be at the top of the retina, because that's where the light will come first. However, the photoreceptors are actually right at the bottom of the retina, underneath all of the other cells, the bipolar cells and the ganglion cells, which are just, their function is just to transmit the electrical signal along. So it's kind of upside down from what you would expect. So that's the first thing to get over. So now the next layer on top of the retinal pigment epithelium, therefore, is actually going to be the photoreceptor layer. So I'm going to draw two different types of photoreceptor, a rod cell and a cone cell. These are the two different types of photoreceptor that you have within the retina, although as we'll see there are 3 different types of cone cells, but as far as the picture is concerned, all of those 3 different types of cone cells look the same, so I'll just draw one rod cell and one cone cell, but I'll come back to the fact that there are 3 different types of cone cells. Okay, so let me begin by drawing a rod. So the reason that some of the photoreceptors are called rod cells and some of them are called cone cells is because the actual portion that is going to transduce the light into an electrical stimulus. In the case of rod cells, it looks like a little rod, it's in a cylindrical shape, whereas in the case of a cone cell, it's in the shape of a cone, an ice cream cone sort of shape. So let me show you this. So I'll start by drawing a rod cell. So here is the portion that's actually going to transduce the light, and here it gets even more extreme because the bits of the rod and cone cells that are actually going to transduce the light, they are right at the bottom and the rest of the rod and cone cells is up higher. So here's the actual rod of the rod cell. Okay, and inside this rod it's going to have these membrane bound discs here, these discs of membrane which are going to contain loads of special receptors for light, but we'll come back to that later on. So this is the actual rod of the rod cell, basically. Okay, it's a cylinder with lots of these discs, so imagine a disc of membrane. Okay, imagine a sphere of membrane, so we can imagine a sphere like so, and phospholipid bilayer this membrane is, and then you squash that down into a disc and you stack these discs on top of one another inside this rod here, and that's what a rod actually consists of. Okay, so those are called the discs inside the rod. Okay, so then what we're going to do is the rod cell will go up like this, so it's got a little connection to a portion here which will contain lots of mitochondria, so I'll just draw these as little black blobs there, so those are supposed to represent the mitochondria. Then we've got another little connection, another little stalk to the bit where the nucleus is, so here is the nucleus, and then it will have an ending here where it will synapse onto and the next cell along, which will be a bipolar cell. Okay, so this is the rod cell then, and this is the portion down here, this actual rod bit, this is the portion that's actually going to transduce the visual stimulus into an electrical stimulus. So I'll outline this entire rod cell in red here. Right, so there is our rod cell. So we can divide rod cells and cone cells, and I haven't drawn a cone cell yet, but we'll see that the same is true of a cone cell, into 2 portions then. The portion is actually the phototransduction area, and this is known as the photoreceptor outer segment. So that's this bit down here. So this is the photoreceptor outer segment, and it's called that because it's more towards the outside of the retina. Remember, this is the outer portion, this is the inner portion. So that's the photoreceptor outer segment, and then the other portion is the photoreceptor inner segment. So all of the rest of this with the nucleus and the area with the mitochondria and this little terminal up here, those are the inner segments, the photoreceptor inner segment. And this is specifically a rod photoreceptor. Okay, so there we have the photoreceptor outer segment and the photoreceptor inner segment of a rod cell. Right, so there's a rod cell, that's one type of photoreceptor, and there is just one type of rod cell, so we're going to have a huge number of rod cells on each of our retinae, and they're all going to be identical, so they're all going to do an identical thing. Whereas the other type of photoreceptor, the cone cells, there are different types of cone cells, but they all look the same as far as our picture is concerned. So I'll draw now one of these cone cells. So I'll start with its outer segment here. And its outer segment is the bit that looks different. So rather than having this cylinder with lots of discs inside, instead its outer segment looks like this. Okay? It doesn't have the discs, and you can see that it's gradually expanding like a cone there. Okay, so that's what the cone cell outer segment looks like. It has these continuous invaginations of its membrane like this, and the whole purpose of this is to try and increase the surface area of the membrane, because we need a lot of these special receptors for light that are integral membrane proteins, so they have to have some membrane to sit in. And the way that the rod cells solved this was to just have loads of these discs inside the cell membrane here, inside this rod portion of the rod cell. Whereas the way the cone cell solves this is it just has loads of these invaginations in this way. So two different approaches to getting a huge amount of cell membrane. And you can see that it sort of looks a bit like a cone here rather than like a rod, hence why these are called cone cells. Then it's just exactly the same. So we have a portion where we're going to have lots of mitochondria here, and then a little connection to where the nucleus will be. There we go, and then a terminal up here where it will synapse onto the bipolar cell. So this is a cone cell. Okay, right, so I will colour in the cone cell, I think, in green here. And the next thing that I need to talk about is the fact that we're going to have multiple different types of cone cells. Now, I will come back to how this is actually useful later on. For now, I just want you to accept that there are multiple different types of cone cells. We will come back to how they actually differ and the trichromatic theory of light later on. But I will give you just a slight introduction to that now, actually. So, rod cells, these are important in black and white vision. They're not important in colour vision. They're important in that you can see using the rod cells. So imagine we had a retina just with rod cells. You would be able to see, but your vision would be black and white. All you'd be able to tell the difference between is where areas have bright light and where areas have unbright light. You wouldn't be able to tell the difference between different colours of light. Okay? Whereas the cone cells, they are important for you having colour vision, and in order for this to work you have to have multiple different types of cone cells which have differing sensitivities to light of different colours. Okay, so you actually have 3 different types of cone cells which used to be called blue cones, green cones, and red cones, and clearly this is talking about which colour light they prefer to absorb. However, there is a better name for them, because actually when we study these in more detail, we'll see that red cones don't really actually brilliantly absorb red light. It's more yellowy light that they like. So the better names for them is to call the blue cones the S cones, for short wavelength light cones. Remember, blue light is short wavelength light. So blue cones, we can call those S cones for short wavelength cones. The green cones, remember green is in the middle, so medium wavelength cones, the M cones. Okay? And then finally, what we used to call the red cones, we now call those the long wavelength cones, so we call them the L cones. And I won't underline that in red because they more absorb in the yellowy orange spectrum rather than the red spectrum. So, overall, dotted around the retina, you're going to have these 3 different types of cones: blue cones, green cones, and red cones, or S cones, M cones, and L cones. And they differ not in their appearance, they all look like this, but they differ in the type of photoreceptor that they have in their membranes, and what wavelength of light that is particularly good at absorbing and responding to. Okay, and we'll come back to how the cone cells are going to actually be useful in giving us information about the colour of the light that we're actually seeing later on. For now, just accept that you have these 4 different types of photoreceptors: rod cells, which only come in 1 different type, and then cone cells, of which we have these 3 different types. Excellent. Right, so That's the photoreceptor layer then of the retina. What I now want to show you is the next layer up from that. So these photoreceptors are going to synapse onto bipolar neurons. So what I want to now draw is a bipolar neuron here. So the next layer up of the retina is the bipolar cells. So to draw a simple picture of bipolar cell, and as I'll discuss in a moment when we come on to the topic of convergence, this is a little bit too simple a picture, but for now, as an introductory picture, here we go. And I'll just draw one of these, and it can be synapsing with this rod cell here. So this next cell up in the layers of the retina then, this is called a bipolar cell. And for short, we'll abbreviate a bipolar cell down to a BC. Okay, so I'll colour in the bipolar cell in orange, like so. So here is our bipolar cell, and you can see that the reason it's called a bipolar cell is that it has two processes coming off its cell body. So here's the cell body of the bipolar cell here, and you can see it's got two processes coming off, hence it has two poles, two sides, and hence why it's called a bipolar cell. And its job is to transmit the electrical signal that the photoreceptor has created in response to light onwards. So this will transmit the electrical signal to the bipolar cell, and the bipolar cell will transmit it to the next cell along, which is going to be the ganglion cell. And we'll talk in a lot more detail later on about the nature of these electrical signals. So the next cell along is going to be a ganglion cell. So here is the synapse of the ganglion cell with the bipolar cell. Here is the cell body of the ganglion cell. And then here is the axon of the ganglion cell. And this will go off into the optic nerve, and it will take the message to the central nervous system, as we'll discuss in much more detail later on. Now, as we'll see later on, the ganglion cells are actually the first type of cell in this pathway to use an action potential as the electrical signal. The photoreceptors and the bipolar cells, the electrical signal in those two cells is just in the form of a change in the electrical potential difference across the cell membrane, rather than an action potential. Okay, so here is the ganglion cell, which is often just abbreviated down to the GC. And I'll outline our ganglion cell in blue here, so like so. Right, so I now just want to give you a little bit of terminology about the different layers of the retina, because there are these fancy terms that people absolutely love to use when talking about the retina, such as the inner plexiform layer, the outer plexiform layer, the inner nuclear layer, the outer nuclear layer. So I want to make sure that you know what all of these different terms mean. So, we'll start then with the outer nuclear layer. So, the layer of the retina where the cell bodies of the photoreceptors are, this is known as the outer nuclear layer, often abbreviated down to the ONL. So this stands for the outer nuclear layer of the retina. So when you look at a section of the retina histologically, I've just drawn, one of these series. There will be loads of these. Imagine replicating this loads and loads of times. You'd have loads of these photoreceptor cell bodies in a great big line, and this is a very distinguishable layer. It looks very obvious because you can see the nuclei. So that's why this layer is known as the outer nuclear layer. It's very obvious when you see it histologically. So in terms of my picture, it's not truly obvious why you would want to use these terms. If you actually get the chance to look at a histological slide of the retina, it becomes much more obvious why people created these terms, because these are quite obvious layers of the histology of the retina. So this is the outer nuclear layer then, this layer where all the cell bodies with the nuclei are for the photoreceptors. And because it's towards the outside of the retina, that's why it's called the outer nuclear layer. Then you have this layer which is made up of the terminals of the photoreceptors and these processes, these outer processes of the bipolar cells. This is known as the outer plexiform layer. So that's P. The only thing I'll write here is what the P stands for, which is plexiform. Okay, so this is a layer that doesn't contain any cell bodies. It's just going to contain lots of processes of these cells. It's going to contain the terminals of the photoreceptors and these processes of the bipolar cells, and of course they'll synapse on one another. So that's the outer plexiform layer. Right. Then we have another nuclear layer here, which is where all the cell bodies of the bipolar cells are. This is called the inner nuclear layer. Again, remember that in order to really understand the importance of these layers, you need to see this histologically. Imagine you've got loads of these in a great big line, then you can imagine that it would appear significant if you were looking at this histologically. So INL stands for inner nuclear layer. Then we've got another plexiform layer here, where we've got the process of the bipolar cell, the inner process with the bipolar cell, and this process from the ganglion cell here. That's known as the inner plexiform layer, often abbreviated down to the IPL. So that's the inner plexiform layer. Then we've got another cell body layer, and this is called the ganglion cell layer, so GCL for short, ganglion cell layer. And then finally we've got the layer that contains all of the axons from the ganglion cells, which is known as the fibre layer. So this is the fibre layer up there. So those are all the different layers of the retina, given their old histological names. So as I say, really to understand why someone would want to create all of these names, you have to see a whole picture of the retina, where you've got this repeated loads and loads of times, and then these layers become much more obvious. Okay. So the next thing I want to talk about, so this has now shown us the basic circuitry of the retina. So we've got photoreceptor, then bipolar cell, then ganglion cell. And I've drawn this just for this rod photoreceptor, but the same thing will be true for the cone photoreceptors. So the cone photoreceptors will be attached to a bipolar cell, which will then attach to a ganglion cell. Okay, this is the basic circuitry of the retina. Now, the next thing I want to talk about, a really important concept, is the concept of convergence, because what I have shown you so far, this basic circuitry, is not quite true. We need to make something a little bit more complicated, and the bit that we need to make more complicated is that it is not true that every single photoreceptor of the retina has its own bipolar cell, which then has its own ganglion cell. The ratio is not 1 to 1 to 1. Instead, you have convergence. So I want to discuss this topic of convergence, and this will bring us quite nicely onto a discussion of the fovea as well. Okay, so convergence then. So in each eye, there is approximately 100 million photoreceptors. So go on to either eye, right or left eye, have a look at the retina, and in there will be 100 million photoreceptors, which are abbreviated down to PRs. The question is then, how many ganglion cells are there? Well, from what I've told you at the moment, your guess would be that there would have to be 100 million ganglion cells as well, because every photoreceptor, as far as I've told you so far, has its own bipolar cell, which then has its own ganglion cell. That is wrong. Instead, there are 1 million ganglion cells. So that means that there are 100 times more photoreceptors than ganglion cells. So what does that have to mean? There has to be convergence. Approximately 100 photoreceptors must send their electrical signals to just one ganglion cell on average. Okay, so let's talk about how this is going to work. So this is particularly apparent for the rod cells. The rod cells in particular converge. So the way that it will actually work is that you will have lots of rod cells in a little region of the retina. So let's just draw these out here. So I'm drawing now a much more basic picture. So here are lots of rod cells shown here in red. Okay, so these lines are just going to represent rod cells. So lots of rod cells now will end up synapsing onto just one bipolar cell. So I'll draw my bipolar cell here. So here can be its cell body, and then it will have just two processes like so, but then this outer process here, it will split into loads of different little bits, which will then synapse with all of these photoreceptors, like so. Right, so here is my bipolar cell, and I should have coloured it in orange rather than changing the colour, but hopefully you get the message. So here is a bipolar cell, these are rod photoreceptors, and then what will happen is loads of bipolar cells will synapse onto just one ganglion cell. So I'll try and draw this. So let's now have another one of these setups. So here are loads more rod cells, and here is another bipolar cell here, synapsing onto all of these rod cells, like so. So again, I'll colour in these rods in red here, and we'll have that bipolar cell again in purple. And now lots of bipolar cells are then going to synapse onto a single ganglion cell. So I'll now draw my ganglion cell up here in blue. So here can be its cell body, and again it will have one process coming off, and here is its central process that's going to the central nervous system. But then this one will divide up, and it won't just divide up into two, you know, you'll have more of these. Okay, so here, this is the ganglion cell. So the overall message here is that no, it's not the case that a single photoreceptor synapses onto a single bipolar cell which then has its own ganglion cell. Instead, loads of photoreceptors will all end up sending their message to just one ganglion cell. Okay, so convergence is particularly high for rods. So a huge number of rods will all converge on the same ganglion cell in the way that I've shown you here. It's not so drastic for the cone cells. You do get convergence of the cone cells, so just like this you'll have multiple different cone cells all going onto a single bipolar cell, and then multiple bipolar cells going onto a single ganglion cell, but it's not as extreme, so you don't have as many cone cells corresponding to one ganglion cell as you do for rod cells. In addition, you must understand that when we have convergence of cone cells, those will be cone cells that correspond to the same colour. So it wouldn't do at all to have cone cells of different colours, of different types, converging onto the same bipolar cell, because then we lose information about colour. So when you have convergence of the cone cells, it will be of cone cells of the same type. So loads of, for instance, blue cone cells might all converge onto the same bipolar cell, and then loads of bipolar cells that have all got blue cone cells synapsing onto them might converge then onto a single ganglion cell. So we don't want to lose the information about the different colours that comes from the different cone cells, so you'll only have convergence of the same type of cone cells. In addition, you would never, for instance, have convergence of rods and cones. So you wouldn't have a bipolar cell that has both rods and cones synapsing onto it, because again that would lose information about colour. Right, so I just want to say a few words about how this changes, this alters the sensitivity of the different types of photoreceptors. So I've talked about how convergence is particularly high for rod cells, so you will have a huge number of rod cells all going on to the same ganglion cell. So a huge number of rod cells in a patch of the retina. So if I draw the retina here, so let's say this is the retina viewed using an ophthalmoscope. Let's say we've got a little patch of retina here, and we'll imagine that all of these rod cells in that little patch are now going on to a single ganglion cell. This means that the rod system has a very high sensitivity, okay? And the reason that I say that is because you don't actually need that much light to be falling on this region. You don't need very intense light to be falling on this little spot in order for that ganglion cell to start firing, okay? The reason being that if you activate any of the rod cells in there, you'll get the electrical activity being transmitted to the ganglion cell. So because you've got loads of rod cells, if any of them get activated, that's going to potentially give rise to an electrical signal propagating down the ganglion cell. And for that reason, you don't actually need that intense light falling on this spot, because of the fact that you've got a huge number of rod cells all converging on this single ganglion cell. Whereas imagine if you just have one rod cell going on to a single ganglion cell, then you would need very intense light, because you need light hitting that single rod cell in order to get an electrical signal going down the ganglion cell. So because of the huge convergence of rods, you get very high sensitivity, but you lose visual acuity. So sensitivity is high, but acuity is low. Now what do I mean by acuity? Acuity is all about being able to see in detail, being able to distinguish between two lights that are coming from very near spots. And of course this means that acuity is going to be low, because if we get the ganglion cell corresponding to this spot activated, we don't know where the light actually struck. It could have struck any of the photoreceptors in this spot. So that means that we can't distinguish between exactly where that light came from. It could have been from any of the spots in the visual field that would have put light onto any of this portion of the retina. So that's why we have low visual acuity, whereas if you imagine if we had one photoreceptor going on to one bipolar cell going on to one ganglion cell, we'd have much higher visual acuity, because when the ganglion cell transmitted an electrical signal, we would know that that meant that a specific photoreceptor had been activated, and we'd be able to trace exactly then where in visual space that visual stimulus had come from, whereas because we've got a huge number of photoreceptors over a large region of the retina, all converging on that single ganglion cell, we don't know which of those was activated, okay? And therefore we don't know exactly where that visual stimulus came from in the visual field. Of course, we'll have a good idea, but we won't know as well as we would if we didn't have this convergence. So the rod system then has very high sensitivity but low visual acuity, and this is the reason that the rod system is very good at seeing under low light conditions, so under what we would call scotopic visual conditions. So scotopic visual conditions is like at dusk, so when it's almost dark but you can still see, so when the sun's just going down and everything's starting to get dark but you can still just about see things, that's your rod system that's doing that, and you'll know that you see in black and white. And the reason is that the rod photoreceptor system, it can't distinguish between different wavelengths of light. All it can distinguish between is different intensities. Okay, so under scotopic vision you use your rod photoreceptor system because it has very high sensitivity, so it can respond to those low light levels. But visual acuity is going to be lower, and also you're going to have black and white vision. And the fancy word then for black and white vision is monochromatic vision. Okay, where all you've got is the different intensities of the light rather than the different wavelengths of the light. Okay, so you use the rod photoreceptor system under scotopic conditions, which are where you've got very low light levels. In contrast, the cone system, in the cone system we have much lower convergence, so we have far fewer cones of a certain type converging onto a single ganglion cell. What does that mean? That means that the sensitivity is much lower, okay? Because now, in order to get an electrical signal propagating up that ganglion cell, we have to be getting light hitting a much smaller number of photoreceptors, which will therefore require more intense light. Okay, so that's why the cone system is far less sensitive. Therefore we use the cone system only under what we call photopic visual conditions. And photopic visual conditions is during broad daylight, basically. So when the light conditions are very good, we can use the cone system, but we can't use it under the scotopic conditions because it's just not sensitive enough. They just won't be being activated, basically, and that's why you don't get colour vision during the night time. In addition, the cone visual system is going to have higher visual acuity in general because of the fact that you have lower convergence, and therefore when you get a ganglion cell activated or firing an electrical signal of some sort, and we'll come on to the nature of these electrical signals later on, you will know that that means that the light has struck a smaller portion of the retina, and therefore you can more precisely say where in the visual field that light was coming from, what sort of angle in the visual field that light was coming from. So acuity is going to be higher for the cone visual system. Now what I want to talk about is the specific part of the retina known as the fovea, which is the part where visual acuity is highest. Okay, so let me talk about this then now. So the fovea then. The fovea, we've already been through it. It's a little pit in the retina, right at the back of the retina, in the centre of the macula lutea. And the reason that the fovea is a pit is because the retina is actually much thinner at the fovea than it would be everywhere else. So let me draw a picture of this. So here's the retina, and then here is the fovea. So we've got this pit right at the back of the eyes. And the reason it's far thinner is that it doesn't have all of the other cell layers above the photoreceptor layer. So it doesn't have the bipolar cell layer, and it doesn't have the ganglion cell layer. So let me just put in dashed lines here where the different layers are, like so. Okay, so let's say that this is the photoreceptor layer of the retina, here in green. Okay, then above that we've got the bipolar cell layer, here in orange, and then above that we've got the ganglion cell layers. So this is why the fovea is a pit, because you don't have the bipolar cell layer or the ganglion cell layer on top of the photoreceptor layer at the fovea. So this is the fovea here, and I'll remind you that the fovea has a diameter of around 1.5 millimeters. So why do we not have these cells on top of the photoreceptors here? Well, these photoreceptors are still innervated. They do still have bipolar cells synapsing with them. It's just that their bipolar cells are splayed to the side. So they're bipolar cells that innervate them. So a photoreceptor here in the fovea, its bipolar cell will be over here, moved to the side. So everything's sort of been parted to the side. And again, the ganglion cells will be up here, parted to the side. And the reason for this is so that the light that is going to be coming to the fovea doesn't have to go through all of the other layers of the retina before it actually gets to the photoreceptor. So this, if you like, is the better design. Okay, another thing to say about the fovea is that it majorly contains only cone cells. So cones are present in the fovea, whereas there are very few rod cells within the fovea. Rod cells and cone cells will be present in the periphery, the non-foveal portion of the retina. So this is non-fovea retina. And here you'll have a mixture of rods and cones, whereas just in the fovea you'll majorly just have cones. The final thing to say about the fovea is that it's the place where the visual acuity is absolutely the best. And the reason that the visual acuity is the best at the fovea is that it really does have no convergence. So it is the case that a single cone photoreceptor will synapse onto a single bipolar cell, which will synapse onto a single ganglion cell. So the ratios are 1 to 1 to 1 here. Okay? And that means that you really are going to be able to see most in detail using this portion of the retina, because if one of the ganglion cells becomes activated, you know exactly which photoreceptor must have been activated, and therefore you can trace in the visual field the angle where that light came from very precisely. Hence you can see most in detail using the fovea. This is why if we want to see something in detail, we make sure that our fovea is looking at it. So when we want to look at something in detail, we make sure that the centre of our eyes are looking at that area, are aligned with that area. So for instance, if I want to read this, I'll make sure that my eyes are directly looking at this. I won't try and read it whilst looking at something else. So if I was to look over there, for instance, I wouldn't try reading this. Instead, what I'd do is I'd move my eyes so that I'm directly looking at this, and the idea then is that light from this area will be focused onto the fovea, and there will be the highest visual acuity, which is what I need in order to be able to actually interpret this, because elsewhere I won't have high enough visual acuity to read this. And that process of moving your gaze so that you're fixating on this is known as fixation or foveation, because you're fixating your fovea onto this area. So fixation or foveation. Right, so that's when you move your eyes so that you're using your fovea to see something that you want to look at in detail. So the fovea has the highest visual acuity because there is no convergence at the fovea. Everywhere else there is convergence, particularly of the rod cells. The rod cells have a huge amount of convergence, whereas the cone cells have much less convergence. Okay, right, so that's the basics of the structure of the retina. In the next video, what we'll begin to discuss is the process of phototransduction, so how the rod and cone cells are actually going to turn a visual stimulus, turn light, into an electrical signal.